You've been taking magnesium completely wrong, not because of the supplement, but because of what you eat with it. Coffee, spinach, cereal, even healthy foods can reduce magnesium absorption dramatically. Here are the 6 biggest mistakes doctors rarely explain. Hi, I'm Dr. William Lee. If you're eating this common food, taking magnesium glycinate right afterward could actually work against your body. Here's what you need to know. Watch the video until the end so you don't miss any important information. If you enjoyed the video, please leave a comment, share it with others, subscribe to the channel, and follow for more upcoming content. So, without further ado, let's begin today's topic. Magnesium glycinate, also called magnesium bisglycinate, is a chelated form of magnesium bound to glycine molecules. Chelation refers to the process by which a mineral becomes attached to organic compounds, such as amino acids. In this case, magnesium is attached to glycine, which may improve absorption and stability within the digestive tract. Glycine itself is a non-essential amino acid involved in neurotransmission, collagen synthesis, glutathione production, detoxification pathways, and sleep regulation. Glycine acts as both an inhibitory and excitatory neurotransmitter, depending on the receptor context and neurological environment. The chelation process protects magnesium from interacting prematurely with compounds in the digestive tract that may inhibit absorption. This may contribute to magnesium glycinate's relatively high bioavailability. Consumers and practitioners often prefer magnesium glycinate for sleep support, stress management, anxiety reduction, muscle recovery, long-term supplementation, sensitive digestive systems, migraine support, cardiovascular maintenance, PMS, symptom management, and general magnesium replenishment. Unlike magnesium citrate or magnesium oxide, magnesium glycinate is less commonly associated with laxative effects at moderate doses. Magnesium glycinate is one of the most widely discussed and increasingly utilized forms of magnesium supplementation in modern nutrition science, preventive wellness, functional medicine, fitness recovery, sleep optimization, stress management, and digital health communication. Interest in magnesium supplementation has grown substantially because magnesium is involved in hundreds of biochemical reactions throughout the human body, influencing muscular function, nervous system regulation, cardiovascular balance, metabolic energy production, glucose regulation, electrolyte stability, neurotransmitter synthesis, bone mineralization, and cellular signaling. Among the many forms of magnesium available in commercial supplements, magnesium glycinate is often considered one of the most bioavailable and gastrointestinally tolerable forms. The combination of magnesium with glycine, an amino acid that participates in neurotransmission and collagen synthesis, contributes to the supplement's reputation for gentleness and calming effects. Consumers frequently seek magnesium glycinate for sleep support, anxiety reduction, muscle recovery, migraine prevention, stress resilience, menstrual support, cardiovascular maintenance, and overall wellness optimization. At the same time, the online conversation around magnesium supplementation has expanded dramatically across social media platforms, blogs, podcasts, YouTube channels, wellness newsletters, short-form video platforms, influencer marketing ecosystems, and health education communities. Viral content about magnesium often includes dramatic claims, oversimplified advice, anecdotal transformations, supplement stacks, sleep hacks, recovery protocols, detox claims, and biohacking trends. Some of this content is evidence-based, while other material exaggerates outcomes or spreads misinformation. A comprehensive understanding of magnesium glycinate therefore requires exploration of both biological science and communication strategy. The scientific side includes absorption mechanisms, digestion, transport, nutrient interactions, dosage timing, deficiencies, contraindications, population-specific considerations, and clinical evidence. The communication side includes digital marketing psychology, educational framing, trust building, audience segmentation, platform algorithms, misinformation management, ethical wellness branding, and viral health content design. This document explores magnesium glycinate from multiple perspectives, including nutritional biochemistry, pharmacokinetics, physiology, supplementation strategy, dietary compatibility, food interactions, timing optimization, population considerations, research interpretation, and long-form content strategy for educational and viral communication. Magnesium, as a biological mineral, Magnesium is one of the most essential mineral elements in biological systems and plays a fundamental role in sustaining life across plants, animals, and microorganisms. As the fourth most abundant mineral in the human body and the second most abundant intracellular cation after potassium, magnesium is involved in hundreds of biochemical reactions that maintain cellular structure, energy metabolism, nerve conduction, muscle contraction, genetic stability, and cardiovascular function. Although it is often overshadowed by minerals such as calcium and iron in public discussion, magnesium is indispensable for the survival and proper functioning of nearly every living organism. The biological importance of magnesium begins at the cellular level. Every living cell contains magnesium ions that contribute to the stability of membranes, proteins, and nucleic acids. Magnesium is a cofactor for more than 300 enzymatic reactions in the human body, including those responsible for synthesizing DNA, RNA, proteins, and ATP, the universal energy currency of cells. Without magnesium, cells would lose their ability to produce energy efficiently, transmit signals, and maintain normal physiological balance. This mineral also regulates ion channels, influences hormone signaling, and supports the structural integrity of bones and teeth. In the natural world, magnesium occupies a central position in the process of photosynthesis. The chlorophyll molecule, which enables plants to capture sunlight and convert it into chemical energy, contains magnesium at its core. Through this role, magnesium indirectly supports nearly all ecosystems on Earth by facilitating the production of oxygen and organic matter. Consequently, the mineral is not only crucial for human health, but also for the continuation of life itself. Historically, magnesium was identified as a distinct chemical element in the 18th century. The name derives from Magnesia, a district in ancient Greece where magnesium-containing minerals were first discovered. Over time, scientific research revealed the extraordinary biological significance of magnesium and its contribution to metabolic health. Today, magnesium deficiency is recognized as a widespread nutritional issue linked to numerous chronic diseases, including hypertension, diabetes, osteoporosis, cardiovascular disorders, and neurological dysfunctions. The study of magnesium as a biological mineral encompasses multiple scientific disciplines, including biochemistry, physiology, nutrition, medicine, agriculture, and environmental science. Understanding magnesium requires examining its chemical properties, dietary sources, absorption mechanisms, physiological roles, interactions with other nutrients, and effects on disease prevention and treatment. The mineral's influence extends from microscopic cellular reactions to whole organism physiology and ecological sustainability. Modern lifestyles and dietary habits have increased interest in magnesium nutrition. Processed foods, depleted soils, stress, alcohol consumption, and certain medications contribute to inadequate magnesium intake in many populations worldwide. Researchers and healthcare professionals now emphasize the importance of magnesium-rich diets and supplementation to support public health. Moreover, magnesium-based therapies are increasingly explored for their therapeutic potential in conditions such as migraines, depression, arrhythmias, and metabolic syndrome. Magnesium exists naturally in many foods, particularly green leafy vegetables, nuts, seeds, legumes, and whole grains. Water sources and mineral-rich soils also contribute to magnesium intake. Once consumed, magnesium is absorbed mainly in the small intestine and distributed throughout the body, where approximately 60% is stored in bones, while the remainder is found in muscles, soft tissues, and body fluids. The kidneys regulate magnesium balance carefully, conserving or excreting the mineral according to physiological needs. The complexity of magnesium metabolism reflects its widespread biological functions. In nerve tissue, magnesium modulates neurotransmitter release and protects against excessive neuronal excitation. In muscles, it works alongside calcium to coordinate contraction and relaxation. In the cardiovascular system, magnesium helps maintain normal heart rhythm, blood pressure, and vascular tone. In skeletal tissue, it contributes to bone mineralization and influences calcium metabolism. In immune function, magnesium supports inflammatory regulation and cellular defense mechanisms. Research into magnesium biology continues to expand rapidly. Scientists are investigating how magnesium influences gene expression, mitochondrial function, oxidative stress, aging, and chronic inflammation. Emerging evidence suggests that magnesium status may affect mental health, sleep quality, insulin sensitivity, and immune resilience. These discoveries highlight the mineral's importance not only in preventing deficiency diseases, but also in optimizing long-term health and well-being. Magnesium glycinate absorption mechanisms. Magnesium glycinate is one of the most widely discussed and frequently recommended forms of magnesium supplementation because of its reputation for high bioavailability, gentle gastrointestinal tolerance, and broad physiological significance. To understand the absorption mechanisms of magnesium glycinate, it is essential to first appreciate the biological importance of magnesium itself, the chemistry of glycine, the structure of chelated minerals, the anatomy and physiology of the digestive tract, and the molecular transport systems involved in mineral uptake and regulation. Magnesium is the fourth most abundant cation in the human body and the second most abundant intracellular cation after potassium. It participates in more than 300 enzymatic reactions, including energy production, nucleic acid synthesis, ion transport, protein synthesis, neuromuscular conduction, glucose metabolism, and maintenance of cellular membrane stability. Despite its importance, magnesium deficiency is common in many populations due to dietary inadequacy, gastrointestinal disorders, chronic stress, medication use, alcohol consumption, metabolic disease, and agricultural depletion of soil minerals. The effectiveness of magnesium supplementation depends heavily on the chemical form administered because different magnesium salts vary dramatically in solubility, intestinal transport behavior, stability, and tolerability. Magnesium glycinate is a chelated compound formed when magnesium is bound to glycine molecules. Glycine is the smallest amino acid and possesses unique physicochemical properties that influence magnesium absorption. In magnesium glycinate, magnesium is usually attached to two glycine molecules through coordinate covalent bonds, creating a bisglycinate chelate. Chelation protects magnesium ions from interacting prematurely with dietary inhibitors such as phytates, oxalates, and phosphates in the gastrointestinal tract. This protection is significant because free magnesium ions can precipitate or form insoluble complexes that reduce absorption efficiency. The chelated structure enables magnesium glycinate to remain relatively stable during transit through the acidic stomach environment and the alkaline conditions of the small intestine. This enhanced stability contributes to greater bioavailability compared with many inorganic magnesium salts, such as magnesium oxide. The process of magnesium glycinate absorption begins in the oral cavity, although only negligible mineral absorption occurs there. Saliva moistens the supplement and initiates dissolution. Once swallowed, the compound enters the stomach, where gastric acid plays a central role in preparing the supplement for absorption. Hydrochloric acid lowers gastric pH to a highly acidic range, often between pH 1.5 and 3.5. This acidic environment increases the solubility of many mineral compounds. In magnesium glycinate, gastric acid helps maintain dissolution while preserving much of the chelated structure. Unlike magnesium oxide, which requires substantial acid-mediated ionization to liberate magnesium ions, magnesium glycinate already exists in a more soluble and biologically compatible form. The glycine ligands stabilize the magnesium atom and reduce the likelihood of insoluble precipitation. Stomach motility also contributes to absorption efficiency. Mechanical churning mixes magnesium glycinate with digestive secretions, increasing surface area exposure and ensuring gradual delivery into the duodenum. Gastric emptying rate affects how rapidly the supplement reaches the absorptive regions of the intestine. Slower gastric emptying may improve mineral uptake by prolonging contact with digestive fluids, though excessively delayed emptying can alter solubility dynamics. Certain factors such as high-fat meals, stress, medications, and hormonal signals influence gastric emptying and may indirectly affect magnesium absorption kinetics. After leaving the stomach, magnesium glycinate enters the duodenum and jejunum, which are the principal regions for magnesium absorption. The small intestine contains highly specialized epithelial cells called enterocytes that regulate nutrient transport. Magnesium absorption occurs through two main pathways: passive paracellular transport and active transcellular transport. The balance between these pathways depends on luminal magnesium concentration, physiological need, vitamin status, hormonal influences, and intestinal integrity. Passive paracellular transport accounts for a substantial portion of magnesium absorption under conditions of adequate or high magnesium intake. In this pathway, magnesium moves between enterocytes through tight junctions driven primarily by electrochemical gradients and solvent drag. Tight junction proteins called claudins regulate permeability and selectivity. Claudin 16 and Claudin 19 are especially important in renal magnesium handling, while intestinal claudin expression also contributes to magnesium permeability. Because magnesium glycinate is highly soluble, it provides a consistent pool of magnesium available for passive diffusion. The glycine chelate may help maintain magnesium in a transportable state until it reaches absorptive surfaces. Active transcellular transport becomes particularly important when dietary magnesium intake is low or physiological demand is elevated. This pathway involves magnesium entry into enterocytes through specialized ion channels located on the apical membrane. Two key proteins are transient receptor potential melastatin channels 6 and 7, commonly abbreviated TRPM6 and TRPM7. TRPM6 is especially important in epithelial magnesium absorption and is highly expressed in the distal small intestine and colon. TRPM7 also contributes to magnesium homeostasis and has broader cellular functions related to ion regulation and signaling. These channels selectively permit magnesium ions to enter enterocytes from the intestinal lumen. The role of glycine in magnesium. Magnesium glycinate is one of the most widely discussed and frequently recommended forms of magnesium supplementation because of its reputation for high bioavailability, gentle digestive effects, and broad physiological benefits. At the center of this compound is glycine, a simple yet biologically powerful amino acid that plays a far greater role than merely attaching itself to magnesium for transport. Glycine contributes to the stability, absorption, tolerability, and overall functional impact of magnesium glycinate within the human body. Understanding the role of glycine in magnesium glycinate requires a close examination of both magnesium and glycine individually, their biochemical partnership, their physiological interactions, and the reasons this compound has become a preferred option for many healthcare practitioners and consumers seeking effective magnesium supplementation. Magnesium itself is an essential mineral involved in hundreds of enzymatic reactions throughout the body. It participates in energy production, muscle contraction, nerve signaling, DNA synthesis, protein synthesis, cardiovascular regulation, glucose metabolism, and electrolyte balance. Despite its importance, magnesium deficiency remains widespread because of dietary insufficiency, stress, gastrointestinal disorders, medication use, and modern food processing practices. Symptoms associated with inadequate magnesium intake can include fatigue, muscle cramps, sleep disturbances, anxiety, irregular heartbeat, headaches, weakness, irritability, and impaired metabolic function. Because magnesium is required in so many systems, even marginal deficiency may influence overall well-being. The challenge with magnesium supplementation lies not only in replacing magnesium stores, but also in ensuring the mineral is absorbed effectively and tolerated comfortably. Many forms of magnesium exist, including magnesium oxide, magnesium citrate, magnesium chloride, magnesium sulfate, magnesium malate, magnesium taurate, and magnesium glycinate. Each form differs in elemental magnesium content, absorption characteristics, gastrointestinal effects, and therapeutic applications. Magnesium glycinate is especially valued because it combines magnesium with glycine in a chelated structure that improves absorption and reduces the likelihood of digestive discomfort. Glycine is the smallest amino acid in the human body, characterized by a simple structure consisting of a hydrogen atom as its side chain. Although glycine is categorized as a non-essential amino acid because the body can synthesize it, this classification can be misleading. Under conditions of stress, illness, rapid growth, inflammation, injury, or metabolic demand, endogenous production may not be sufficient to meet physiological needs. Glycine serves numerous functions throughout the body, including participation in collagen synthesis, neurotransmission, detoxification, antioxidant production, immune regulation, and metabolic balance. When glycine binds with magnesium to form magnesium glycinate, it acts as more than a passive carrier. The chelation process creates a stable molecular complex in which magnesium is attached to glycine molecules through chemical bonds. This structure protects magnesium as it travels through the digestive tract. Without proper stabilization, magnesium ions may interact with dietary compounds such as phytates or oxalates, potentially limiting absorption. The glycine chelate shields magnesium from premature interactions, increasing the likelihood that it reaches intestinal absorption sites efficiently. One of the primary reasons glycine is important in magnesium glycinate is its contribution to bioavailability. Bioavailability refers to the proportion of a nutrient that enters circulation and becomes available for physiological use. Magnesium oxide, for example, contains a high percentage of elemental magnesium, but tends to have relatively low absorption. In contrast, magnesium glycinate often demonstrates superior absorption because the glycine component facilitates transport across intestinal membranes. Amino acid chelates may utilize amino acid transport pathways in addition to mineral absorption mechanisms, potentially improving uptake. The digestive tolerance associated with magnesium glycinate is another major advantage linked directly to glycine. Some forms of magnesium, particularly magnesium oxide and magnesium citrate at higher doses, can attract water into the intestines and produce laxative effects. While this may be useful for constipation, it can be undesirable for individuals seeking long-term supplementation without gastrointestinal distress. Glycine's chelation helps reduce the amount of free magnesium in the gut, lowering the likelihood of diarrhea, cramping, or irritation. This makes magnesium glycinate a preferred option for people with sensitive digestive systems. Glycine itself possesses calming properties that complement the physiological actions of magnesium. Magnesium is often associated with relaxation, stress reduction, and sleep support because it influences neurotransmitter activity and nervous system regulation. Glycine also functions as an inhibitory neurotransmitter in the central nervous system. It interacts with glycine receptors and modulates neuronal excitability, contributing to relaxation and sleep quality. The partnership between magnesium and glycine may therefore create synergistic effects, particularly in individuals dealing with stress, anxiety, insomnia, or nervous tension. In the spinal cord and brainstem, glycine acts as an inhibitory neurotransmitter by allowing chloride ions to enter neurons, decreasing the likelihood of excessive firing. This inhibitory effect helps maintain balance within the nervous system and prevents overstimulation. Magnesium contributes to neural stability by regulating NMDA receptors, which are involved in excitatory neurotransmission. Excessive activation of NMDA receptors may contribute to anxiety, excitotoxicity, migraines, and neurological stress. Together, magnesium and glycine help support balanced neural signaling. Sleep support is one of the most commonly discussed uses of magnesium glycinate, and glycine plays an important role in this effect. Research suggests that glycine may improve subjective sleep quality, reduce the time required to fall asleep, and enhance daytime performance after sleep restriction. Glycine may influence thermoregulation and circadian rhythms, helping the body transition into restorative sleep states. Magnesium contributes by supporting muscle relaxation, nervous system calmness, and hormonal balance. The combination of magnesium and glycine may therefore offer broader sleep-related benefits than magnesium alone. Another important aspect of glycine's role in magnesium glycinate relates to collagen production and connective tissue health. Glycine is a major component of collagen, the structural protein found in skin, joints, tendons, ligaments, cartilage, bones, and blood vessels. Adequate glycine intake supports tissue repair, elasticity, and structural integrity. While magnesium glycinate is not primarily marketed as a collagen supplement, the glycine component still contributes amino acid support that may complement overall tissue maintenance. Glycine also participates in the synthesis of glutathione, one of the body's most important antioxidants. Glutathione protects cells from oxidative stress, detoxifies harmful compounds, and supports immune function. Magnesium is involved in many enzymatic processes related to antioxidant defense as well. Therefore, magnesium glycinate may indirectly support cellular resilience through both magnesium-dependent enzymatic activity and glycine-dependent glutathione synthesis. The liver relies on glycine for detoxification reactions, including conjugation pathways that help process toxins, drugs, and metabolic waste products. Glycine participates in bile acid formation and supports metabolic balance within hepatic tissue. Magnesium similarly contributes to liver function by participating in enzymatic reactions involved in energy metabolism and detoxification. Their combined presence in magnesium glycinate creates a compound that supports multiple overlapping physiological systems.